Welcome to lecture 8 of recombinant DNA technology. In our last lecture, we are going through some of the techniques that are required to purify the proteins. We are continuing with these techniques. Today, we will be discussing about the polyacrylamide gel electrophoresis, which is also known as sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE. In the research lab, it is widely known as Western blots. This technique is mainly used to separate the proteins and their identification and detections after separating using the polyacrylamide gel. So proteins can be negatively neutral or positively charged, and SDS, which is an anionic detergent, and if you can recall, we use this SDS extraction of DNA. So that provides a uniform negative charge to all over the proteins. And in the sample, so that helps to separate the protein mixtures. So SDS-PAGE or SDS is also used as a lysis buffer in addition to other reducing agents like beta-mercaptoethanol or dithiothiotol, and then hitting the proteins to dissociate the polypeptides and then run into the gel. So the gel electrophoresis is exactly the same as we have seen for DNA, but in case of DNA, we use the agarose, and for protein identification or detection, we use the polyacrylamide gel. So the amount of SDS bound is generally sequence non-specific or independent, and it's proportional to the molecular weight. So when an SDS polypeptide complex migrates in an electric field, this is normally proportional to the relative size of the polypeptide chain. So in these techniques, there are mainly 3 steps to be considered. Firstly, the proteins are separated through this SDS-PAGE, and then it has been transferred from gel to a solid support membrane, which is a nitrocellulose or PVDF, and then detecting the proteins into that solid membrane using antigen antibody complex and visualizing them mainly with chemiluminescence technique. Similar to the DNA separation or DNA analysis, the lower molecular weight protein travel faster and the higher molecular weight slower. And a molecular ladder has been used. In this case, is a molecular weight ladder rather than the length, which we have been seen in DNA analysis. So that is giving an indication about the molecular size of the protein of interest. So as we have told the process that the protein has been first lysed, and then in the lysis buffer, SDS is always used with, as I said, that beta Markov ethanol and dithiothiool, either one of them, as a reducing agent that denature the proteins, and then running the gel to identify the proteins. And then the protein has been transferred to a membrane, which is a nitrocellulose or PVDF, which is also called the polyvinylidene fluoride membrane. And then using a blocking agent to cover the spaces on the membrane which is not bound by the proteins. That means the blocking agent actually covering all the areas on the membrane where the proteins from the cell is not present. The reason for that is in the next step, when we use the specific antibody that has been bind to a specific site or the protein of interest, so there wouldn't be any unspecified binding of our antibody. And that's why the blocking step is very important. So the protein of interest in this case is the antigen, and a specific antibody has been used to combine and form a complex of antigen antibody, so that in the next step we can detect our protein. So there are two antibodies has been used. One is a primary antibody and another one is the secondary antibody. The primary antibody, which is mainly bind to the protein of interest, and then the secondary antibody, which has the affinity for the primary antibody and which is also having a specific chemicals that is called a horseradish peroxide, HRP, which actually use in the next step when the protein has been visualized by chemiluminescent method. So that is in a nutshell the Western blot for detection of proteins using the SDS page. So antibody can be a monoclonal antibody or a polyclonal antibody. So monoclonal antibodies, they are more specific to the protein of interest. So they can be highly bind to a specific area of the protein. And they're just a one antibody can be bind. So this is highly specific. And this monoclonal antibody is normally produced in ex vivo method. So they can be produced first into the animal. It can be rat, rabbit, or any other mouse. And then from there, that antibody has been extracted and produced in a cell culture, which is in vitro. So that's why it's called ex vivo. So some of the part is inside the cell and some inside the living organism, and then some of them is just in vitro. One is in vivo and then in vitro. So that's why it is very specific, and the production of the monoclonal antibody especially when batch-to-batch consideration, the variation is very less. On the other hand, the polyclonal antibody is actually the antigen is injected into the host organism, and the host organism then develops all kind of antibodies for that exact protein, for that specific protein. And that all antibodies has been extracted. So the polyclonal antibody has also a high specificity, and it is good when there is a less amount of protein concentration to be detected. So the protein concentration, the concentration of the protein of interest, that should be the right way. The concentration of the protein of interest is lower, then we use the polyclonal antibody, because this might have 3 or 4 antibodies for a single specific protein, and it can be detected easily. But the problem is, as this is in vivo production, there is always a variation, batch-to-batch variation, depending on the host organism and the first batch might be given a more specific antibodies, the second, the same organism, maybe rat. For the second batch, it might not. So there is a batch-to-batch variation in polyclonal antibody. And the secondary antibody is always have the specificity for the primary antibody with a chemical, mainly used the horseradish peroxide. That horseradish peroxide has been interact with the chemicals, the detection chemicals, and emit a blue light during the detection. And that has been, the detection has been done in X-ray film. But these days it can just detect it into the spectrophotometer. So the detected protein can be visualized into the screen. But many labs, they prefer to use the X-ray film to develop the proteins. Next is a isoelectric focusing. So isoelectric focusing or isoelectric point is a point where, or at a pH level where the net charge of a protein is 0. So as there is a different combination of amino acids into the protein, so there is overall charge may be positive or a negative. And below the pH, and at the pH level below their isoelectric point, the proteins normally exhibit the positive charge, and above at negative charge. So when there is a pH which is equal to the isoelectric point, they form a Zwitter ion, and that has a net charge is 0. So this isoelectric focusing method is also used to separate the proteins depending on their isoelectric point. SDS phase is more separating the protein depending on their molecular weight. This is the difference between their isoelectric point. So the pH gradient has been used for this, and when the electric current has been applied, so if the protein is a positively charged, then they are more pulled towards the negative end, and the negative charged protein goes to the positive end. But at some point, when at the pH, when the protein, the isoelectric point is equal, they don't move anymore. So their movement has stopped because they don't have the charge to move along either the positive end or the negative end. So this isoelectric point and SDS page both used together to have a two-dimensional gel electrophoresis. So the first is isoelectric focusing has been used for separating the proteins based on their isoelectric point, and then that separated protein has been run through a gel SDS page to further separate that proteins according to their molecular weight. So this is particular experiment is important when you have a two or more proteins which have a very close isoelectric point values. And this isoelectric point, as you saw in the picture, so it is more like a rotating by the gel is placed in a 90-degree angle. So when the isoelectric focusing is like this, but it has been then produced as a horizontal or vertical, just the opposite way, the experiment has been done in isoelectric point and in SDS page. So we have this cartoon is just giving a bit of explanation how the proteins has been separated first using an isoelectric focusing point, and then using that separation gels, use the SDS page to separate according to their molecular weight. So these are very common tools, especially the SDS page, which has been regularly used into the labs for identification and detection of protein. So this brings to the end of this chapter. As like the previous lectures, I have been included some of the videos. So just go through the videos. I hope they makes the thing more understandable for you. And if you have any questions regarding this lecture or any of the previous lecture, I am happy to discuss during the discussion.